For the better part of a century, paleoanthropology was dominated by a single geometric shape, the tree. It was a tidy, comforting model. A thick central trunk represented the success of Homo sapiens, while other hominins, like the Neanderthals, were relegated to dead branches, evolutionary failures that snapped off and fell into the sediment. We told ourselves a story of replacement. We believed that when our ancestors expanded out of Africa, they moved into a world of strangers, outcompeted them, and replaced them without biological interaction. But that tree is a simplification that no longer fits the data. Since the advent of high-throughput next-generation sequencing, the silence of the fossil record has been broken by the precision of molecular phylum genetics. The model that has emerged, championed by researchers like Rebecca Ackerman, is not a tree, but a braided stream. It is a model of reticulate evolution, a chaotic network where lineages diverge, evolve in isolation, and then merge again. In this narrative, the Neanderthals did not simply go extinct. Through a process of assimilation, they became part of the stream that flows through us. To understand how this happened, we must look beyond the caves of Europe. We must turn our attention to the specific engines of this evolution: a waiting room on the Persian Plateau, a climatic corridor along the 55th parallel, and a ghost lineage that has haunted our genome for 2 million years. Our re-examination begins in the Altai Mountains of southern Siberia, the easternmost frontier of the Neanderthal range. Here, deep within the genome of a single female, geneticists identified a sequence that defied the standard taxonomy. Approximately 3% of her DNA originated from a human group that was neither Neanderthal nor Denisovan. For years, this was categorized simply as a super-archaic signal, a statistical anomaly. However, a 2020 analysis by Rogers and colleagues utilized site pattern frequency analysis to finally propose an identity for this ghost. The data is most consistent with Homo erectus. The implications are profound. The modeling suggests that roughly 700,000 years ago, the ancestors of Neanderthals and Denisovans expanded into Eurasia and encountered a population of Homo erectus, a lineage that had diverged from the main human trunk nearly 2 million years prior. They did not merely coexist, they interbred. This suggests that the Neanderthals were never a pure species in the Victorian sense. By the time they migrated west into Europe, they were already a hybrid lineage, carrying the genetic memory of the very first hominins to leave Africa. They were biological archives, preserving the code of a species we thought had vanished eons before. But if the Neanderthals carried ancient ghosts within their blood, they were also masters of their distinct geography. For decades, the connectivity between European Neanderthals and their Siberian cousins remained a mystery. How did related populations bridge 3,000 kilometers of hostile terrain? In late 2025, a study by Piggott et al. Provided the answer through a bone fragment catalogued as Star 1, found at the Starosale site in Crimea. Using zooms, zooarchaeology by mass spectrometry, researchers identified this fragment amidst thousands of animal bones. The proteomic fingerprints confirmed it was human, but the DNA told the story of travel. Star 1 was not genetically linked to the nearby European Neanderthals, but rather to the Chagirskaya lineage in the Altai, thousands of kilometers away. Piggott's team reconstructed the mechanism of this movement. They identified a habitat suitability corridor running precisely along the 55th parallel. During the warmer interstitials of the Ice Age, this latitude did not support forest, but rather a biomass-rich steppe tundra. It was a superhighway for megafauna. The evidence suggests these Neanderthals were specialized equestrian hunters. By tracking herds of horses and bison along this specific latitude, they maintained a transcontinental network linking the Black Sea to the mountains of Mongolia. They were not static cave dwellers, but highly mobile predators navigating a specific climatic niche. While Neanderthals navigated the northern steppe, the ancestors of non-African Homo sapiens faced a different reality. The archaeological record presents a chronological gap. We know modern humans left Africa roughly 60,000 to 70,000 years ago, yet we do not appear in the colonization records of Europe or East Asia until approximately 45,000 years ago. Where did this population reside for 20,000 years? In 2024, a study led by Vallini proposed a solution to this missing years paradox: the Persian Plateau Hub. Combining paleoclimatic modeling and genomic reconstruction, the data indicates that during the arid and cold conditions of marine isotope stage 4, the Persian Plateau, encompassing modern Iran and Afghanistan, served as a crucial refugium. While much of the northern hemisphere was inhabitable, this plateau supported a stable ecosystem. It acted as a demographic incubator. Here, the ancestors of all Eurasians differentiated, developing the basal genetic signature that underpins modern populations in both Europe and Asia. But this hub was not isolated. To the west, in the Zagros Mountains, lay the territory of the Neanderthals. It is highly probable that the Persian Plateau served as the primary contact zone. The 1 to 2% of Neanderthal DNA found in all non-African populations today is likely the result of sustained interaction in this specific region, a period of cohabitation that reshaped our immune systems forever. However, the braided stream flows in both directions. We have long focused on what Neanderthals gave to us, but a 2023 study by Harris et al., utilizing diverse African genomes, has revealed what we gave to them and when we gave it. The study detected regions of Homo sapiens DNA within the Neanderthal genome that date back to an introgression event between 250,000 and 270,000 years ago. This creates a significant adjustment to our timeline. Long before the successful out-of-Africa migration that colonized the globe, there appears to have been an earlier, failed expansion. These early pioneers likely reached the Near East or the edges of the Persian hub, where they encountered Neanderthals. They did not replace the archaic population. Instead, they were genetically absorbed. Over millennia, natural selection acted on this hybrid genome. Because the two populations had been separated for so long, genetic incompatibilities likely arose. The deserts of human DNA in Neanderthal genomes and vice versa are evidence of purifying selection, slowly scrubbing these deleterious alleles over time. By 40,000 years ago, the distinct Neanderthal phenotype vanished from the fossil record. Genetic samples from sites like Mezmaiskaya Cave show a population in collapse, suffering from low heterozygosity and inbreeding depression. When the demographic pulse from the Persian Plateau finally released the main wave of Homo sapiens, the remaining Neanderthals were likely swamped by the sheer numbers of the newcomers. But in a reticulate model, they are not gone. We carry their legacy in the genes that regulate our keratin production and, crucially, in the HLA variants that help our immune systems recognize pathogens. We are a mosaic. The story of human origins is not a story of a lonely winner standing atop a tree. It is a story of a river, fed by the durability of Homo erectus, the cold adaptation of the Neanderthals, and the plasticity of Homo sapiens. The bones may be silent, but the blood still speaks.